Oh, yeah, that works. Let's turn that down. I don't know if it's worth it. It's probably worth it. Yeah. Hi, everybody. Good to see you all. Is this mic too loud or okay? We're good? Okay, awesome. So welcome to the second half of BIS 220. Okay, and I'm taking over from Professor Barakat, who, as we all know, is awesome, a great instructor. And I will try to, reach your level. We'll see if I can do it. I'm an associate professor of neurobiology here at USC. I started in 2014. I run a research group in systems neuroscience. We study how the cortex processes sensory information and develop new tools for imaging, neuromodulators like dopamine, acetylcholine, norepinephrine, things that sort of control your emotions and your attention. Outside of that, I've got 3 kids, like to ride a bike, exercise a lot, and I'm really interested in how physiology and organ systems and the body system sort of fit together to achieve like very high levels of performance. So that's sort of like what I enjoy. I'm displaced from the Palisades. I've got this awesome fit. Thank you, Kelly Slater, for the free clothes. But things are going okay. I'll be back there in 6 months, I think. So I want to let you know how to reach me. So you can call me Professor Hires, say Edward Hires, I don't really care. I go by my name. You can come reach me before or after class. If we're going to do extended Q&A sessions after class on Tuesday, it'll take a few minutes to set up. That'll be on Zoom. You're welcome to drop in for office hours anytime. My lab is in Head Co-Neuroscience building, HMB 228. You might want to email ahead of time, though, just in case I'm not there, because I might not be there. Always running around doing different stuff, particularly right now. This term's been crazy. So that's communications. So there's Brightspace. We'll post content on Brightspace. Besides Brightspace, I would say, what's the most useful tool right now for learning? LLMs. If you're not using ChatGPT or DeepSeek, Claude, to sort of accelerate the pace of your learning, you really should try to explore that. And I'm actually working on a little task force we put together to try to figure out how to bring that methodology into the classroom better. But haven't quite figured it out yet, because it's going to require a lot of sort of rethinking about how classes are going to work. But I think by the end of this term, I'll have some document that I'll be able to put out, and you guys can all take a look at it and see if it's helpful. So it's great for studying, right? Like you could throw up a set of slides and they're thrown in and be like, give me 10 questions on this, right? And you're going to be evaluated, of course, on your exam performance. So you need to have that information in your head. You can't just like throw it into an LLM. But, you can use that to develop like study skills and study tools and, give me questions that are relevant to this particular chapter of the book, and it'll spit stuff out. And you can always use that to sort of teach yourself new things if the amount of content that we have is insufficient for you. Okay, so, and, of course, we don't want you using that as a tool for your writing or lab reports or anything right now, just because part of the educational process is for you to learn how to write as well. Okay. Okay. So midterm 2 is coming up. Thank you all for showing up, even though this content today is not on midterm 2, but it will be on midterm 3. So it's very important to show up. So that's next Friday in 3 different locations. That'll be on Brightspace, posted on Brightspace soon. And then, one of the most common questions is, what do I need to know for the test? I'd say read the chapters that are in the syllabus for each section. Make sure you sort of focus on concepts that are in the lecture topics. I'm going to try to make sure every question on the exam is at least touched on within the lectures or in the lecture slides. Okay. And we'll have practice exams that are real exams from prior years that I'll put up with answer keys, so you'll be able to sort of get a feeling for what my exams are like. They're fairly similar across the sections. Sometimes we share questions across sections between the different professors, and of course every sort of class is sort of curved to its own average in case there's differences in difficulty on exams between different sections, so nobody gets an advantage for being in the easy class or in the hard class. Okay. All right, so I think it's really important to know the concepts, because that's going to help you organize facts, and the more concepts that the more deeply you understand the concepts, the better you're going to do. All right, any questions on any of that stuff? We're good. All right. Cool. So I guess, what do I expect? I expect you to, ideally, if you want to do your best, read the textbook material prior to class, try to identify any areas you don't really understand fully, so that you can then sort of formulate questions about that content, engage in discussion. Be happy to answer questions in the middle of class, raise your hand, and we'll address it. And then together, I think putting all those things together are going to help you build a foundation for understanding how the body works. So this half of the course is different from the first half, right? This first half is all cell biology, sort of understanding what's going on inside the cells, fundamental principles of how the machinery of the cell works. But now we've got to sort of take that out and put it together into much greater organ systems, right? So we're going to go through most of the major organ systems of the body, right? This next 2 weeks is going to be cardiovascular system, respiratory system. So how do we basically exchange gas with the environment? How do we get rid of carbon dioxide, take in oxygen, and deliver that to all of the different tissues in the body that need it, right? We're warm-blooded creatures. We burn a tremendous amount of oxygen every minute that we're alive. If you go without oxygen for a few minutes, you're going to die. So much of our system, much of our body is designed to facilitate that delivery of oxygen. So, right. So I think in any sort of section of the course, you might want to ask these sort of broad-based questions is, what life needs are going to have to be filled or why do we even have this organ, right? Or this system, right? There's got to be some life need that's going to be fulfilled by that system, right? Otherwise, we wouldn't have it. It would have been evolved away, unless it's like the appendix. Then, it's always useful when you're thinking about what life needs to be fulfilled. I gotta admit that person. Okay. I think it's important to also think about, why is the organ system designed the way it is, right? There's going to be some physical principles, the physics or the chemistry of it, that actually guides how the organ has been designed to now successfully achieving its overall goal. Okay. And we want to understand how that system works in health. That's most of the course. We can sometimes, we don't do it too much in this course, certainly more so in the book, is that we might look at variations that are adaptive in other species. How is the heart designed differently for like a frog or a fish than a mammal? How are the lungs different in a bird versus a human? These are sort of adaptations that sort of, because different creatures will have different sort of, they work in different environments. You want to have maybe a different style of design of the organ system to achieve the goal of it. And then, of course, how many people here are pre-med? Wow. Yeah, there we go. Okay. So we want to know what happens when things go wrong. Disease, right? Many of you want to try to figure out how to cure disease, how to keep people healthy, and understanding how the system works. What's in health, if something gets tweaked and goes wrong, right? If you get a tumor in your adrenal gland, what's going to happen, right? How is that going to change the growth of your body plan and stuff? So always those sort of mutations or little diseases tell you a lot about how the system is designed and how it works. All right. So I think for the cardiovascular system in particular, you can just look to the Tour de France. Okay. This is sort of like the pinnacle of cardiovascular design of these two people, right? We've got Tadej Pogačar in front, Jonas Vingegaard in the back, here trying to chase them up a giant mountain. Okay. And this is an incredibly difficult task that is going to require the delivery of the maximum amount of oxygen over long periods of time. Huge amounts of aerobic respiration. This is how, so they're going to try to achieve their goals. And if you look at how the body sort of adapts to stress, you can see how people in the Tour de France over the years have tried to maximize their performance, right? How do you make red blood cells grow? What do red blood cells do? What's their primary function? Yeah. Carry oxygen via hemoglobin, right? So how could we maximize our performance? With maximize the amount of red blood cells that we have, right? So initially, we had like Lance Armstrong a long time ago, he injected himself with EPO, erythropoietin, which is the drug or the molecule inside the body that enhances the production of red blood cells. Okay, that got banned. Then people started to infuse their own blood. They take blood out, put it in a box over here for like a month, and then during the tour, they infuse blood back in to try to increase the red blood cell count. That will allow them to achieve performance. That got banned. So then now they've moved on to new things. Maybe we don't even know what they are because the performance keeps going up. Well, carbon monoxide is a new thing that people are trying to breathe carbon monoxide to sort of disrupt the bind to their hemoglobin and block oxygen from hanging on, make it more difficult for the body to sort of deliver oxygen during training, and then stop doing the hemoglobin and the homeostatic adaptive response of the body to try to make more hemoglobin when you're sort of under carbon monoxide, then gives you a boost later on of having more hemoglobin once you're off this carbon monoxide. That just got banned, I think, a month ago. So anyway, all of these things, right, are things that are going to relate to the performance of the body, and they all are there are different ways to trigger adaptations to particular kinds of stresses. Okay, so what are like two kinds of stresses that might be present, say, at the top of Mount Everest, or if you wanted to hike up to Everest Base Camp? What are you going to have to deal with here? Yeah, lack of oxygen, right? Like this is, so probably what you might want to have to live at a high elevation for a few weeks to have some sort of adaptive response to increase your red blood cell count. And that will allow you to have more red blood cells, and you'll be able to then exercise and walk around much better up this level. This is why people do the high altitude training as well. Is there anything else that might be? Yeah. Cold temperature, right. So you can adapt to a certain extent to cold temperature. I think that Wim Hof guy that's always like sitting in an ice block likes to do it, or the Huberman's always doing his cold ice baths. So you can have some sort of body adaptation to that, but of course, we are also intelligent creatures that are tool users, right? And we dominate the world with our technology. So we've learned how to wear clothes, to create clothes. Okay, so we can have some sort of adaptation that is technological to these sort of extreme environments as well. And the sort of overriding theme of all of this is that the body wants to maintain a certain level of constancy within many aspects of it, right? So you want to maintain temperature in a narrow range to make sure that your body's processes are optimized. All the chemistry that goes on within your body really does best at around 98 degrees Fahrenheit, okay, or 37 C, okay? And so if we look here sort of in the cells, right? In the cells of the body, you're going to have within the animal's internal environment, we're going to have very small internal fluctuations to keep those cells happy and doing things they want. So not just temperature, but also things like pH, how acidic the interstitial fluid around it is. You're going to want to have enough oxygen there. You're going to want to get rid of waste products like carbon dioxide. You're going to want to need an energy substrate that's available. Glucose, you need to be there. All these things are going to have to be maintained within narrow levels. So within the animal's internal environment, we're going to have homeostatic mechanisms that regulate every one of these sort of little things that can vary, okay, that can impact the effectiveness and the health of the cells in the body, okay? And then we also have an external environment, and these sort of internal environments are going to be influenced, of course, by our external environment, how much oxygen is there, how much temperature is present, is it hot or is it cold? So this was a concept of the homeostasis was actually created by Claude Bernard, who was one of the great biologists of the 19th century. And he discovered that the liver controls blood glucose levels and also articulated this concept of homeostasis first. So homeostasis simply means the body staying the same. Okay, that's the Greek roots. Okay, so let's look at another example here. So how are we going to maintain blood glucose levels? So it's very important to have a particular level of blood glucose, a fairly narrow, I mean, it can maybe vary by a factor of 10, okay? But it's very important to have blood glucose levels within this range, within a safe range, because although parts of the body, like the muscles, can store glucose, the brain has very little glucose storage, okay? So if your blood glucose goes down below 40 millimoles per liter, or 40 millimoles per liter, you're going to start feeling sick. Below 20, you're going to have to go into a coma, can potentially die. Now, the good news is that we have mechanisms by which we can regulate that overall level of glucose. One of the primary mechanisms is the release of insulin, which is going to bring glucose into storage mechanisms within the cells of our body. Okay, so here's a plot here, and I think on any plot, it's very important. The first thing you ever want to do, okay, and this is read the axes, okay? And if there's no axes on a plot, it's not labeled, and there's no like tick marks. Like when people presented in my lab, I'm like, this is my #1 pet peeve. You're making a plot, make sure you are labeling it, because otherwise nobody knows what it is. But if you read the axes, you're going to be able to see it. So here we are. So on this axis is time, and this axis is the concentration of blood glucose, and the axis is how much insulin we have. Okay, so the red line is related to the blood glucose level. The blue line is this blood serum insulin level. And what we can see is that over time, the blood glucose, sort of varies with time, and it goes up and down. And when does it go up? When we ingest food, right? So breakfast, lunch, and dinner each trigger a large increase in blood glucose. Now, almost within a minute or two of starting that ingestion of food, we also see a concomitant increase in this blue line, which is the blood insulin levels, okay? So the blood insulin levels are going to react and tell the body, and sort of signal to the body, signal to the liver and other cells in the body to take glucose in, because we have a sudden spike in these glucose levels. We don't want it to go too high, okay? That we want to sort of maintain and seize that blood glucose, and then be able to release it later after sort of digestion is complete. So that's one example of a homeostatic mechanism where we are going to try to provide a negative feedback to bring something that is going out of the range that we want it to be in back into a narrower range. And so all homeostatic control systems, so that's what we would call a homeostatic control system. All of these are going to share a particular three parts. First, we've got to have a receptor. Okay, we have to have something that is going to detect a change in a particular variable, right? So we have levels of carbon dioxide receptor that's going to in our brain that's going to sort of figure out how long has it been since we last took a breath. And it's going to have a very powerful influence on trying to trigger us to breathe again and flush out our carbon dioxide. So we have to have a receptor. We have to have a control center, okay, that's going to take information in from that receptor or sets of receptors. Sometimes it'll have more than one receptor type, okay, and then direct a response that is going to then try to affect the response through the effector to bring levels back in line to where you want them to be. So another example here is in the body, but also in your dorm room. Okay, it's the temperature, right? So with the temperature control, we have some sort of sensor, okay, and we have sort of set point here that we want to sort of have the room at, some room temperature, okay, 20 degrees Celsius. Okay, if the temperature goes to a high, the thermometer sensor is going to recognize that. It's going to tell the control center to turn off the heater. Okay, and then we're going to have a drop in the room temperature, sort of back to baseline. On the other hand, if the temperature comes too low, okay, the thermometer or receptor will sense that, tell the control center to turn on the heater, and you're going to turn up the heat. The room temperature is going to increase, and you're going to bring it back into this optimal level. Okay. Now, it doesn't perfectly hold it there, right? You have to have some fluctuation that goes, a little too high or a little too low in order to trigger that response. Otherwise, your air conditioning would be going on and off every 20 seconds. Okay? Probably not very efficient. So that sort of range in which it's considered stable and you're not triggering a response is called the hysteresis. Okay, so that's sort of the level of variation that you can maintain around your set point. Okay, so what we can see here, though, is that in both of these cases, whether you're going too high or you're too low, what happens is the feedback mechanism tries to bring it back to that baseline. If it's too low, it tries to bring it up. If it's too high, it tries to bring it down. So this is a concept called negative feedback. It's a negative feedback regulation because it is resisting the direction of the deflection, right? It's trying to bring it back to a normal level. And negative feedback mechanisms are going to be common all over our body systems. They're much more common than positive feedback systems because most of what our body wants to do and most of what our organ systems want to do is maintain this homeostasis. There are exceptions, okay? Positive feedback mechanisms, and you'll see that oftentimes when you want a specific event to occur, okay? Like the delivery of a baby. Okay? There we have a lot of positive feedback mechanisms with oxytocin, pressure that are going to actually cause uterine contractions and expel the baby. So there's a couple of examples like that. You see it also. A lot of aspects of reproduction are like that. Okay. Any questions on homeostasis? All right. Oh, go for it.
Does homeostasis involve positive feedback or only negative?
Does homeostasis involve positive feedback or only negative feedback? Homeostasis is generally going to be negative feedback. Okay, almost always. I can't think of an example where positive feedback would maintain homeostasis, but maybe there is one that I'm not thinking of. But almost every system that's homeostasis is going to be almost exclusively negative feedback regulation. So negative doesn't mean it just goes down, right? Negative is just meaning it's going to resist the direction of deflection, right? Any other questions? Okay. So I want to spend like, I don't know, like 30 minutes just talking about general principles, okay, and general physical principles that are going to govern so many of our system designs in our body. And without getting into like the nitty-gritty of individual organ systems. Just what are some basic principles that are going to affect so many different things? Okay, and what does our body really need to do? Okay, one of the most important things it needs to do is deliver stuff. Okay, it needs to deliver nutrients to the right place. You've got to deliver gas to the right places. You've got to get rid of waste to the right places. Okay, it needs to produce energy, right? We have to create ATP somehow. We've got to drive all those cycles that you've probably learned about to do all of our biochemistry. Well, we're going to need to have some sort of food that comes in, and we need to convert that to glucose, and we're going to need to burn those fuels. But of course, we're also going to have there's going to be waste products from that, okay? And we're going to have to figure out how to get rid of those waste products, maybe through the kidney, and then and also through things like the stool, right? So we have waste. What are those waste products? How do we get rid of them? That's another important aspect of our organs. Okay, now to the physical principles. There's only a few that I really want to highlight because they're so important. The first one can be illustrated by this. Okay, we've got something small and we've got something big. Okay, and you might say, okay, so let's imagine these are two cubes. Okay, and this cube right here has an area. Let's say the side of length of this cube is 1 unit, right? So what's the overall surface area of the cube? What's the surface area of one face? 1 times 1. Okay, so 1, square units or whatever. And we've got 6 sides. So that's 6 times 1, that's 6 units. Okay. What about this? Okay, let's say 1, 2, 3, 4, 5. I hope that's 10. If it's 10 long, though, well, then the surface area of one side is not going to be 10 units. It's going to be 100 units because it's 10 times 10, right? And then putting up all 6 faces together, that's 600. So by increasing sort of dimension along one axis by a factor of 10, the actual surface area is going to increase by the square of that. Okay. However, that is not what happens with the volume of the object, right? In this case, with the volume of the object, the volume is just going to be one side times one side times one side. So that would be 1 unit. The volume of this is going to be 10 times 10 times 10, 1,000 units. So it is the volume, as you get bigger, the volume increases at a much faster rate than the surface area of something. And this is very important in understanding how different scales of different organisms, as well as different parts of our body, are going to have different designs to deal with this difference in scaling of area versus volume. And you can see this is sort of illustrated in the body plans of these two organisms here. Got this little water skimmer, we've got a giant elephant. What do you see that's different about these two creatures in their body? The size is definitely different. This one is bigger than this one. Right, so now, so we've got the size different. What about the shape? What are some differences in the shape? Yeah, you want to say something. I know you've got your hand like this. You're ready.
The bug is like much slimmer.
The bug is, the legs are a lot slimmer, right? Yeah, legs are a lot slimmer. Got those thick elephant legs right there. So why might that be a difference in the plan? Well, probably because the strength of, since the weight, the mass of the animal is related to the volume of the animal, right? As you get bigger, your mass is going on into the cube of your sort of legs. Okay? But the strength of your legs, of your muscles, is only related to the cross-sectional area of the muscle. So that as a muscle gets bigger and bigger and bigger, as a mass gets bigger and bigger and bigger, the muscle has to proportionally increase much more dramatically to support that weight. Okay. I mean, this thing's so light it can't even go underwater. Surface tension. Put an elephant in the water, it's going to sink pretty quickly. So elephant has to have much thicker legs relative to the rest of its body than an insect. Some other aspects, some other differences here too, though. If you're small, right, you have greater amount of surface area for the volume. What might that do to how quickly you exchange materials in your environment? Yeah. One of them might let off more CO2 than the other. So they're going to exchange gases more easily, I think, when you have a greater surface area for the amount of space that you're taking up, right? More surface area for a ratio to volume ratio means exchange is going to be faster because the exchange is the material moving across the surface. Okay. So that could be with CO2, or it could be with a gas. Gas exchange can happen through the skin of these creatures. Okay. It also is important for things like heat and thermal regulation. Okay, so this animal is going to be much more dependent on the external temperatures. Okay. It's going to, because it's so small, temperature can rapidly change, it will rapidly change its body. Here, the elephant, it's got this big body here, okay? Not a lot of surface area for the amount of volume that's coming out of there. Okay. So it's going to have a hard time removing its heat. Especially like African elephant here, it's hot up there. How's it going to remove its heat? Well, it has these specialized sort of ears here. Right? Those ears are really thin, wide flaps that can move them backwards and forwards. They're full of blood vessels. Okay? So this is a way that they can increase the surface area of one part of their body to sort of radiate heat out and regulate their heat in an active way by moving those ears back and forth. Okay, so and these are just constraints on sort of physical constraints on how big you are really impacts how your body plan is going to be designed. I guess, okay. So if we think about that elephant ear, right, what is it actually doing? Okay, how is it dealing with this? How is it sort of trying to solve the process here where we've got too much volume for the amount of surface area that we have to release the heat? Well, it's doing something like this. It's basically taking that volume, okay, and splitting it up into adding an additional surface. Okay. Now we have the same amount of volume, it's the same number of like spheres here, right? Same number of units here. But now by opening up the middle of it, okay, we create two more faces. So now we increase the surface area by I guess 33%, right? So that instead of like having 6 faces, we now have 8 faces, but we have the same volume. Okay, so this is going to increase our surface area by 33%, and that is going to increase our ability to exchange. And you can see this everywhere. This is something that affects the body plan. It affects so many sort of things like lungs. It affects Amazon delivery areas and factories or delivery things where you have a whole bunch of stuff that you want to store it, you want to move it to another place, exchange it somewhere, right? How are you going to design that? You're going to design it so that the large volume of things you have maximizes the amount of surface area access to those things in an efficient way. Okay, so sort of the design of an Amazon warehouse is governed by the same principles of gas exchange with fish, humans, mushrooms, everything, right? We use the same strategy here where you have these invaginations of material that is going to try to optimize for exchange. So this is the gills of either the fish or the mushroom are where the gas exchange is occurring within these things. And this is the area where they try to maximize the surface area to the amount of volume that's going through there. So exchange, we've talked about that. I've used that term a little bit already, but how does exchange even work? What is actually the motivating force that is driving some molecule to move from one location to another location? Let's take a look. This is a force that really operates on a microscopic level. Okay, and so if you put some fat globules of milk, okay, does this play? Let's play. Okay, let's look at what's known as Brownian motion. So this is looking under a microscope. You see, if you look down carefully enough at this, what you see is a bunch of wiggling going around. Okay. Why are these molecules wiggling? This is a still glass. There's no currents moving around. Why are the molecules, why are these little pieces of things wiggling around? Any guesses? What's that?
They're dancing. Sick tunes.
There's sick tunes, yeah. There's no vibration here, so no sick tunes. Yeah.
Yeah.
They're ready to react. What is causing that? Why are they moving? Energy, okay. If we turned up the temperature, do you think they'd move faster or slower? They're gonna move faster. Why? Why are they moving around? What is a fluid anyway? What's it made of? It's made of atoms. It's made of little molecules, right? Little water droplets, right? So when you think about a big cup, I'm taking my cup of coffee, right? I'm gonna swallow, I don't know, hundreds of trillions of molecules of water, right, in one gulp, okay? But when you're zoomed in and looking at very tiny pieces of things, all of a sudden, a fluid is actually behaving as individual molecules that are hitting one another. So the random motion of every molecule within that fluid bouncing around and running into things is actually transmitting momentum to the objects that we're looking at. So those little pigments, right, that we're moving around, that is just the random motion from all the sort of the heat of the energy within that fluid of each individual molecule hitting it. Okay. Now, what's something that you might notice about that? Those pigments. Is there anything different? I think it's easier in the pigment ones. What's different about the big ones versus the small ones? Less movement on the big ones, right? Why might that be? They're bigger. Each one is getting hit by molecules, right? They're getting hit in every different direction at random times, right? Okay. One molecule only has so much mass, though. It only has so much momentum. Okay, so it can move a small object a lot more effectively than a larger object, right? So you're never going to believe who used this observation by looking at sort of pigments in a glass to figure out what the weight of an atom was, predict what the weight of an atom was, and to actually give the first sort of macroscopic evidence that atoms existed. Very famous scientist, most famous physicist in the world, probably. He's famous for the special theory of relativity about how light works, but in the same year he published the theory of special relativity in 1905, he also had this other paper that proved the existence of atoms by looking at the stuff basically in his coffee. Okay, by measuring how much, Einstein showed that by measuring how much each one of these little particles moved, he was able to predict how much mass an individual atom had. Okay, so it's really the momentum transfer of individual atoms that's causing the random motion. And as we can see here, a small molecule is going to bounce around a lot more than a big molecule because it has less mass. Okay, so this process of what's known as Brownian motion, this sort of random motion, is what is going to actually be underlying things like gas exchange. Okay? Small things like a little oxygen molecule here are going to be more effectively going to be able to diffuse quicker than a large sort of macromolecule like this. Okay, smaller things diffuse faster. And there's not very many equations in this course, right? Biology course. There is one, relatively simple one, that is going to govern diffusion. And I really just want you to understand conceptually, like, how does diffusion work? Because it's going to operate very differently on microscopic scales than macroscopic scales. Okay, so with diffusion, okay, what we see is that the distance that you go is going to be equal to the square root of the amount of time that you're allowed to travel for. Okay, so imagine like I'm walking, okay, I'm making a purposeful motion, okay. The distance I go is equal to my sort of rate of speed times the amount of time I'm walking, right? I can walk faster, okay, and I'm going to get somewhere more quickly, okay? But if I go for twice the amount of time, I'll go twice as far. If I go for 10 times the amount of time, I'll go 10 times as far. Okay, that's because I have some direction to my motion. That is not how diffusion works. Okay, diffusion is like the drunk person under a stoplight wandering around and falling over and going back one way, and going back another way, and going back this way, always in motion, right? But never going in a directed flow direction. Okay, so when you start, you're going to end up somewhere else after some amount of time, and on average, the longer amount of time that you are wandering around for, the further away you'll go from the origin. Okay, but because you're also sort of walking backwards and forwards, sort of in a random way, the distance that you travel is actually going to be equal to the square root of the amount of time. Another way to say that is the amount of time it takes for you to go a particular distance in diffusion is equal to the square of the distance. Okay, so what are some actual examples of this? Okay. Really important one is glucose. Okay, glucose is one of the things that we need to exchange, right? And we have to get glucose into our cells, and the cells need to burn it, right? And the glucose has to get around in the cells. At that level, how long does it take for glucose to travel like the length of a cell or the length of a few cells? Okay, well, glucose will travel 100 microns. That's 1/10 of a millimeter. Okay, about the thickness of your human hair, just by random diffusion in about 1 second. Okay, but because of this square function here, to go 1 millimeter 10 times further is not going to take 10 times longer. It'll take 100 times longer, right? So it's going to take 100 seconds to travel 1 millimeter. What happens when we need to go 1 centimeter? That's 10 times more than that. It's going to take not 100 seconds. It's going to take 100 squared seconds. It'll take 10,000 seconds. That's way too long. This is so if you want to deliver something, if you want to move glucose from one place to another place, okay, and those two places are a centimeter away, you're not going to be able to use diffusion to do it. Okay, it's going to take you 3 hours later on average. You're screwed. Okay. On the other hand, if you are trying to diffuse, if you're trying to go inside a cell, let's say 10 microns or something, 10 microns, that's 1/10 of this of 1 second. So, or if it goes 100 microns in 1 second, we're going to go 1/10 of that size. Actually, we'll square that. So it's actually going to be 1/100th of a second. You'll be able to go 10 microns. Okay? If you're going to go 1 micron, it's going to be 1/10,000th of a second. Almost instantaneous, right? So diffusion is incredibly useful and incredibly powerful for transporting things on very small scales, on the orders of microns. It's totally useless for a millimeter and beyond. Okay. So that tells us that when we're in sort of this range of the scale of whatever organism or part of an organism that we're dealing with, sort of in this micron to submicron range, diffusion is going to be the dominant sort of force that's going to be moving things around, transporting. But when we get out to things on the side of and above, we're going to have other mechanisms of transportation, right? We're going to have something that acts like you're walking and can get you there. If you're going to place a car, you just place it long and 4 times as long. Okay? Some directed flow. And that is why we have a cardiovascular system. Right? The cardiovascular system is going to be a huge network of tubes that is going to try to deliver the gas, the glucose, and all those things close enough to every individual cell in your body, such that in that last sort of the last mile, we might say, but the last few microns, right? That delivery, then the diffusion will take over from that point. Yeah. Yeah. Skip that. Any questions about that stuff? Okay, cool. All right, so now those are like 2 principles that are they're so important for all the sort of different organ systems. I just wanted to set them up right from the start. Okay, so now I want to turn to the specifics of the cardiovascular system, and I'm going to open up this water. So let's take a look at this. Okay. Oh man, that sucks. Okay, well, you can see a little bit better now. You can look on your phone or on your laptop too, okay? So here is a structure of the heart with everything but the blood vessels removed, okay? Anybody ever go to those Body Worlds expeditions? No, where they like turn people into plastic and cut them into different pieces? It's pretty cool. I don't know how he got those people to do it, but what we can see here is that there's quite an interesting design here to the heart. Okay, so there's 4 chambers to the heart. There's the atria up high. Okay, and there's these 2 large ventricles down low, and you've got a huge differences in sort of the size of the pipes here. Okay, or the blood vessels. Okay. We have the aorta that's coming off here. This huge output from the heart, and then within the heart itself, within the musculature of the heart, there's tons and tons of really tiny blood vessels. Okay, so we have coronary arteries like this. They're sort of the main deliverers of blood to the muscle. Okay, when you have a heart attack, that's generally caused by a blockage in one of the coronary arteries, which then occludes the flow of blood. This network of very fine blood vessels called capillaries. Okay. Those capillaries are where the walls are thin enough and the meshwork of those different blood vessels into the areas is sufficiently fine that diffusion can move oxygen out and bring carbon dioxide in, exchange that. Okay. So why do we have to have all those blood vessels into the heart? Well, what does the heart do? It's pumping. It's squeezing, 40 to 220 times per minute. Okay, over and over again your entire life. Okay, so it's going to need a lot of oxygen. Its goal is to circulate oxygen throughout the entire body, but it itself is highly energetically demanding, and so it needs to have blood also being delivered to its muscle itself. Okay. So a lot of gas exchange is going to be happening here in these capillary beds into the muscle itself. Do you think there's going to be much gas exchange happening within the aorta? Why not? Right, the wall of the aorta is way too thick, right? So diffusion is not going to be effective, right? We're going to have actually a lot of flow, high speed, blood's going to be flowing out of that aorta, moving past, okay? There won't be a lot of time for oxygen for the blood to sit around there for it to diffuse and exchange. Okay. So this sort of network of tubes that connects everything was really first illustrated by Vesalius in the middle of the Renaissance. And really, if everybody that is, pre-med here, if you want to study the history of medicine, really the Renaissance is where medicine starts to take off, right? So Vesalius sort of looked at the structural architecture of the blood and blood vessels, the arteries and veins. But he and everyone else had the wrong idea about how those blood vessels actually worked. Okay, they thought that the arteries originate in the heart, the veins originate in the liver, and there was tidal flow where these fluids would move in and out, and in and out, backwards and forwards. They're like the tides going up and down on the sea or waves. That is not how it works. Okay. And one of the reasons why it was so difficult to figure out why that's not how it works is because it didn't look like these 2 systems were connected with one another. Because by eye, you could not see those connections. Because the tubes that connect the arteries and veins are so fine, you need the development of the microscope to be able to discover them. These capillary networks, okay, which are going to move blood from the arteries into the veins, are so fine you can't see them by eye. You only see them under a microscope. So as the microscopy was getting developed, and Harvey was able to discover Harvey was able to postulate that there's a different sort of organization where the heart is a pump that flows material around up to the lungs, where gases exchange, and then down to both the liver and the rest of the systemic circulation, then back to the heart. Okay. And postulated, he hypothesized that there was some network of tubes that connected those two systems. Okay. But it was only till Malfigi came around 30 years later that was able to use the Wiedenhock's microscope, look at and actually discover those capillary networks, proving that hypothesis. Okay. So hearts are found in every closed circulatory system. So there's other kinds of circulatory systems, Malpighian tubules and stuff, like in invertebrates, where they don't have to move as much stuff around because they're so darn small. Okay, and they can exchange gas openly with the air. You can read about that in the textbook, but we're going to focus mostly on mammalian stuff in this lecture, at least. So for closed circulatory systems like ours, and like, I think most animals, okay, you're going to have a heart. You're going to have some sort of core area, which is going to squeeze the fluid and increase the pressure, okay? That increase in pressure is going to allow the fluid to flow in a gradient, okay? So this is an important concept here. Why do things flow down a tube? Okay, they always are going to flow from one area of higher pressure to an area of lower pressure. If you take a hose, okay, you screw your hose into a tap outside, okay, turn the tap on, okay, there is a certain pressure of water that is being supplied by the big water tank up on the hill that is sort of using gravity to apply pressure and push down this hose, okay. And so you would have maybe 60 pounds per square inch of pressure here. Okay. And then at this end, you're going to have 0 pounds per square inch of pressure relative to the average pressure around us that the air pushes on us. Okay, so in order for fluid to flow down this tube, okay, we're going to have a pressure gradient. It is going to always be going from an area of higher pressure to lower pressure. Okay. That means there is actually a pressure drop along each segment of the tube. Okay? It doesn't just go from 60 to 0 here, or 60 to 0 here. It goes from like 60 to 59, 58, 57, 56 as you flow down the tube. You can see this. If you were to poke pinholes in that tube, you get greater pressure here. Near the beginning of a tube, it will be flying out faster than at the bottom of the tube. So our circulatory system is a network of much more complicated tubes. Okay. Tubes that branch and branch again and branch again and branch again, and then merge back until we return back to the heart. Okay. So what we can understand from this, though, is that at the beginning, the origin of the flow, okay, at the heart itself, that's where we're going to have the highest pressure. And then in order to make it easy as possible for the blood to return to the heart, we want to have the holding zone for the return of the venous return to have as low pressure as possible, like 0, right? Maybe we want to have 120 millimeters of mercury of pressure on the sort of pumping end and 0 on the receiving end. Okay, and we can actually see that pressure change, then that pressure drop, if we look here across the different steps of the progression of blood vessels throughout our body. Okay, so this is an example from the systemic circulation. There's more than one circulation. In humans, there's the pulmonary circulation that goes to the lungs. Okay, and that's a different circuit than the systemic circulation that goes to the rest of the body. So this is the pressure in the systemic circulation. Okay, and it starts at the heart. Okay, and then travels down the aorta to the arteries. Arteries are branches of the aorta. So the aorta is one big sort of tube that comes off. It branches to the arteries that are still fairly thick, then to arterioles. Okay, the pressure starts dropping into the capillaries. We get even less pressure. One end of the capillaries, though, has higher pressure than the downstream end. Okay, and then by the time we get to the venules, which collect from the capillaries, and then they get pulled into the veins and into the vena cava, which then returns to the heart, we have very low pressure. Okay, and there's also some really interesting sort of feature here, too, right? Like we've got this thing that's going up and down. The pressure is varying, okay? It's pulsing, okay? And that's because of the contractile motion of the heart. We're going to pressurize and depressurize the heart in a cycle. Okay, and pressurize and depressurize the blood in a cycle that's linked to the contractions of the heart. Those pumps, those fluctuations completely go away by the time you get to the veins, and are pretty much gone by the time you get to the capillaries too. Okay, and as you can see, actually, there's very little pressure difference here between the veins and the vena cava. So it's going to be hard for blood to flow actually in the veins. In fact, most of the actual flow that's going to drive blood back into the heart is going to be from contractions of your own muscles, squeezing these sort of low pressure veins and pushing the blood down back to your heart. Okay, so once the blood gets back to the heart, we're going to use our heart to restore the blood pressure and enable recirculation. So let's look at this circuit. So this is the circuit that operates in birds and mammals. Okay, it's a 4-chambered heart. Fish have got a 2-chambered heart. They don't need as much oxygen transfer. Okay, they're cold-blooded creatures. Their metabolism is much lower. Okay, they only have 2-chamber heart, simpler. Frogs are weird. They have a 3-chambered heart. Read about that, and that's kind of because they're amphibians. They can vary how they're going to do gas exchange. But for birds and mammals, we have a 4-chambered heart, and this is because the 4-chambered heart is going to be the most efficient at driving the appropriate amount of pressure to get material in through the lungs versus the overall systemic circulation. Okay, so there's 2 separate circulations here. And let's say the blood is starting here in the right ventricle of the heart. Okay, it's going to be pumped up here into the pulmonary circuit. This pressure is significantly less. It's higher than, of course, it's higher here than the lungs, okay, but it's significantly less than the pressure here, actually, because the sort of path length and the resistance to flow to get through the lungs is less. So we're going to pump from the right ventricle. Okay, you have to imagine you're like standing and looking down at your heart in this image. So the right ventricle is going to get pumped up to our lung capillaries, up to our lungs. Gas exchange is going to occur. You can see this here by the change in color between blue and red. Okay, that is an indication that the hemoglobin is loading up. When hemoglobin gets oxygenated, it changes the color that it absorbs of light that it absorbs, and that causes your blood to look. Your blood doesn't actually ever look totally blue. If you actually cut a vein, the blood doesn't come out blue, but it's a different color red. But when you look through your sort of skin, it makes it look blue. But anything that looks blue in like your arms, right? Those are the veins. Okay. The arteries are going to be really red, okay, because they're full of oxygen. Okay, so the arteries here are coming down. Okay, hold on a second. Okay, so I want to correct myself a little bit there. Okay, so the arteries aren't always full of oxygen. Okay, it depends on which circulation they're in. Okay, artery is something that's moving blood away from the heart. Okay, so the arteries in the pulmonary circulation are going to be blue, deoxygenating blood. Okay. So the deoxygenated blood is traveling up here in the artery, gets through the sort of lung capillaries, gets reoxygenated, and then the pulmonary veins. Veins are things that return the blood to the heart. Okay, the veins come down here, we're fully oxygenated, and we maintain that oxygenation through the passage and the pumping of the left ventricle out into the systemic circulation. We maintain that a level of blood oxygen all the way till we get to the systemic capillaries of the body, where then we have gas exchange that takes place. We unload the oxygen, reload up with carbon dioxide. Okay, that's going to travel back up to the right atrium of the heart and then into the right ventricle in a loop. Okay, so what are some design principles here? We have this fully divided ventricle here. Okay, so we have 4 chambers because that's going to prevent the mixing of the oxygen-rich and the oxygen-poor blood that's coming back. In a 2-chambered heart, those things mix, and it makes the system less efficient. Okay, because you're always pumping up some partially oxygenated blood back to the gills or whatever, and then you're sort of diluting your oxygen level when it's getting pumped out of the systemic circulation. That's not the case here. We're going to have fully deoxygenated blood going up to the lungs and fully oxygenated blood that stays fully oxygenated until it gets down into the systemic circulation because of this divider here between the ventricles. Okay. So this is going to support high metabolic loads and endothermy. That means a warm blood creature. Okay. Now, one thing about this, okay, is that the output of the right side of the heart is always going to have to be equal to the output of the left side of the heart. Okay, because whatever you have to make space for the material to move around. Okay, and since there's so much more resistance to flow throughout your entire body, so there's a lot more paths that you have to go through, right? More friction along it. You're going to have higher pressure coming out of the left ventricle to push it through the systemic circulation than to push it through the pulmonary circulation. Okay. Now let's sort of look a little bit here. This is another sort of, let's see here. So I think I've pretty much already said most of these things, but let's sort of, what else do I want to say here? Okay, a few more things I want to highlight. Okay, so this pulmonary circulation, okay, we're pumping out of the right ventricle, okay? It's going to split, okay, into both lungs when it comes back, okay? It's going to pass through what's known as the sinoatrial valves, and then through these semilunar valves as it passes from the left atrium to the left ventricle, and then into the systemic circulation, which is going to split as well up into the head and down and the forearms, and then down into the rest of the body, okay, before it returns via the inferior vena cava. The superior vena cava is going to be what brings blood back from the head. Now, this is the sort of overall architecture, but there are a few examples where there's things called portal veins, which are going to sort of branch off from this main circulation and deliver specialized blood flow to particular areas. Really important area to deliver blood flow to is the liver, because the liver is where much of our metabolic transformations take place. Okay, so if you eat whatever food you eat, right, and it gets absorbed, okay, you need to turn it into a whole variety of different nutrients and proteins and fats and things like that, right? The liver is basically the factory that's going to have all the biochemical processes that are going to transform whatever you ate into things that your body actually needs. Okay, so you want to have a lot of blood flow into the liver. Also, the kidneys are going to be where we excrete and filter out a lot of our waste. So 25% of the actual blood flow is going to get sort of shunted off and delivered to the kidneys. And we'll talk about, we have a whole sort of week that we're going to go into talking about the kidneys. Okay. So what does the actual cycle look like? You have to design the heart in a way so that you're going to be able to squeeze and pressurize some chambers of the heart, okay, while at the same time relaxing other chambers to allow an area for the blood to come back via the venous return, right? Okay, so, and these successive squeezings and relaxations are called systole for the squeezing and diastole for the relaxation. And let's look at sort of this cycle. We can think about, like, this is a loop, okay? And we could start the loop wherever we want, but we will start it here at atrial and ventricular diastole, because that is where all of the chambers of the heart are relaxed. And that lasts for about, it depends on how fast your heart is beating, okay? But let's say your heart is beating at 70 beats per minute, okay? So I'm sort of typical level here of a awake walking around person or sitting, taking notes. Okay, so we start in atrial and ventricular diastole. That means both the atria, which is the receiving area, which has thin walls, it's very flexible. It's called an atrium. Think of it like an atrium in a building. It's a place, it's just a space, right? A nice space for things, people to walk through or for blood to flow into, okay? So the atria can sort of relax and stretch as blood returns into the atrium. And then if the ventricle is also in diastole, blood can continue to flow into the ventricle and sort of partially fill it up, okay? And that's great. It's partially filling it up, but it's not really stretching that ventricle out a lot. In order to maximize the amount of blood that's in the ventricle before the ventricle itself squeezes, we are going to trigger systole in the atria first, okay? What is that going to do? That is going to provide, it's going to cause a contraction of the right and the left atria, and that will start to increase the pressure in those areas. What's that going to do? That's going to cause the blood to try to move away from that area, right? Move down a pressure gradient. Okay. Well, one area, if the ventricles are relaxed and the atria are contracting, well, the blood's going to try to move into the ventricles, okay, because they're going to move down a pressure gradient. The ventricles aren't squeezing until they get, you start to fill them up, sort of the elasticity and the resistance of that sort of ventricle will start to increase the pressure in the ventricle until they sort of equalize. Of course, it could also go backwards, right? It could go back down the veins. The thing is, veins have valves in them. If you run your fingers across like your veins, you'll find these little sort of lumps along them. And those are little valves that have flaps that will open only in one direction. So if you push, if your muscle squeezes, okay, that will sort of squeeze the vein, and the blood will try to exit that area of the vein, but it can only go in one direction because when it flows back a little bit of ways, it's going to run into one of these valves, which will snap shut. But if it's going in the other direction back towards the heart, that valve is designed to open up in response to that pressure. So that pushes it linearly that way, and that is why the blood is not going to flow backwards into the inferior and superior vena cava because of these valves that are not actually in this diagram. But there are other valves here. So these atrioventricular valves here, okay, these AV valves, so named because they're in between the atrium and the ventricle, okay? These are going to have some flaps that open up unidirectionally and allow the blood to flow in, okay? And then, so that sort of atrial systole, ventricular diastole lasts for about 100 milliseconds, okay, a tenth of a second. Okay. Then we have ventricular systole and atrial diastole. So the ventricles are going to squeeze hard, start pushing blood up into the lungs as well as down into the systemic circulation. And then simultaneously, that's going to occur with the atrium relaxing to provide a space for the blood that's coming, back up from that's getting pushed down to flow back into. Okay. So we're going to have a drop in pressure in the atria because they're relaxing and a giant increase in pressure in the ventricles. That's going to open these semilunar valves and push the blood up into the pulmonary artery and the aorta. So this is a cycle of systole and diastole. Okay. So in the cycle, what we have is a cardiac output. The cardiac output is how much blood are you actually moving from the cycle in a minute, okay? How can we calculate that? Well, we need to know how fast is our heart beating, how many times per minute for the beat, that's our heart rate, you can measure that, just count it. And then we're gonna multiply that by the volume of blood that is expelled through each ventricular systole. Okay. That might be, and that can change, that's called the stroke volume, can change depending on how sort of hard your heart is beating and how much you're relaxing and squeezing. But you can get up to, let's say, a typical amount would be 70 milliliters per beat, 5 liters per minute. If we multiply 75 times 70 milliliters, we're going to get 5 liters per minute. So that's a little bit over a gallon per minute of blood circulating throughout your body. Okay. By the way, 5 liters is the amount of blood that you probably have. Okay, or you probably have, right? So it's all dependent on size, but the total amount of blood you have in your body is much less than you might think, right? It's like, I think, you read like, oh, your body's like 80% water or something. Yeah, sure, but like most of that is inside your cells or in your interstitial fluid. The blood is actually less than 2 gallons of blood within your body, between really 4 to 8 liters, depending on your body size. Okay, so basically circulating at rest, you're circulating all of the blood in your body through the system every minute. Now, if you're exercising, what's going to happen? Your heart rate goes up, your stroke volume goes up. Okay, you're going to dramatically increase your cardiac output. And why is that? It's because you need to move oxygen to the muscles that are exercising, right? And you need to get rid of all that, the metabolic waste products, right? We got to get rid of that carbon dioxide. Okay, so you've got to increase the pace at which you're flowing material throughout the blood body systems. So you could go up to 30 liters per minute. Now, how does that actual squeezing take place? Like, these must be some really weird cells, okay? And they are. The squeezing takes place by cardiomyocytes. These are cells that are automatically contractile, okay? They are what's known as a syncytium, which is a development where you have a lot of different cells, and then during development, they fuse together, okay? They have many nuclei now from their originating cells, and they now work in tandem with gap junctions that are going to allow electricity to travel and ions to travel across them. Okay? So they will work to squeeze and pull together. Okay. Now you can take these. This is what this is what your cardiac muscle is made of. It's going to squeeze. It's going to sort of contract rhythmically, autonomously, without any other thing. It just has these ion channels in it, and it has calcium that flows in it. And we'll learn all about sort of muscle contraction near the end of the course. But essentially, it has these rhythms that are intrinsic to the cell itself that cause it to squeeze and relax on its own. In fact, you can take out the cells from a heart, okay, these cardiac myocytes, put them on a little dish with like PDMS, little sort of flexible things, lay it down there, and the little flaps will start to like contract themselves and move like this. This is from Megan McCain's lab here at USC, which is, you can imagine now, maybe if you wanted to study a drug, screen for drugs that might impact the rate at which the heart beats, for example. Instead of putting it into a human first or an animal first, you could just put it on this dish and see if it changes how fast it flaps. Okay. Now, the thing is, if each one of these cardiac myocytes, these syncytiums, are contracting independently from one another, okay, they're all contracting, but they're doing it at different paces or different timing, you're not going to be able to have an effective heartbeat. You're just going to seize up. Nothing's going to happen. Okay, we need to have some sort of organizing method for which the for which these cardiac myocytes can coordinate the contraction and relaxation differentially across the different ventricles. Okay. And this is going to be coordinated by electrical signals that are sent down Purkinje fibers. Okay, so these are basically fibers that can conduct electricity, okay, and deliver electricity to certain points within the heart muscle in a coordinated way at different timings. Okay, the way that this is going to operate and why they can pass electricity is because electricity is simply the movement of charged ions, so positive or negative ions, okay, in a particular direction, and those ions can pass through these little pores that are formed between neighboring cells here within the Purkinje cells, and they will pass ions through these gap junctions. So where does those electrical signals originate, and where do they get delivered? Okay, so the whole heart muscle will contract on its own. It will beat on its own. Okay, in order to coordinate the relative timing of the systole and the diastole, though, we want to have one set of cells that beats and controls the pace. Okay. It turns out those set of cells tend to beat at a slightly faster pace than all the other cells within the heart. So these cells within the heart maybe want to beat at like 30 beats per minute. Okay. That's what they'll do naturally. But if an electrical signal comes in and kicks them a little bit, then they'll beat faster. They'll do what they're told. Okay. And the thing that's going to be doing the telling is the sinoatrial node. Okay. Why is it called this, or SA node? Why is it called this? It's because it's in the blood sinus, which is basically a sinus is just a area for blood to pool. Okay. In the atrium. So the sinoatrial node is some component here within the atrium. Okay. That is going to be a set of cardiac myocytes that beat at a slightly faster pace than everything else and are controlled via nerves and hormones to regulate the speed of that beat. Okay. So when this pacemaker goes off, it's going to cause a wave of electrical signals to travel across the atrium. Okay. There are conduction mechanisms for that to take place, and that's going to cause the muscles within each atria to contract in a coordinated manner, which then squeezes the blood into the ventricles. Okay. And then that signal is not going to be able to propagate into the ventricles themselves, though. There's an insulating layer between the atria and the ventricles. It can only pass via the Purkinje fibers. Okay. And those Purkinje fibers originate right here in the atrioventricular node. So when this wave of electricity hits this atrioventricular node, it starts to excite that node, but there's a little bit of a delay that's built in there. Okay. And then when this AV node finally gets excited, it's going to fire off a wave of electricity. It's going to travel down this fiber, and this fiber is going to be insulated until it gets to the tip, the apex of the heart, in which case then you're going to have little branches that come off that are no longer insulated. Yes. Yes, the heartbeat is because muscles get shocked. Yes. Your thinking is because your brain is getting shocked in a very coordinated, complicated way, right? So your talking is because your muscles in your larynx are getting shocked. Okay, so what happens here is we have this wave of electricity that's going to come down here. Okay, and then this area is where it first allows to get leaked out into the cardiac muscle. Okay, and then from there it will spread through the rest of the ventricles. Now, why might we want it to? Why not just let it pass like a wave all the way down? It wouldn't do anything. You can push the blood this way into it. We want to push the blood out. We want to squeeze the tube of toothpaste from the bottom, not from the top. Okay? We want to get as much out as possible on our squeeze. And so that is why we have this sort of complicated arrangement of fibers here that come out here. And so the sort of initial, if you're looking at electrocardiograms, the sort of initial depolarization of the electrical signal you see here is from the sinoatrial node. And then this bigger pulse is actually from the ventricles contracting. So how do we control that heart rate? We need to be able to have a way. This is a homeostatic mechanism now. We have to have negative feedback to always have a heart rate within a useful range. Okay. And we can do that through nerves and hormones and other environmental factors. So let's look here at the sort of nerves. Okay, so we need to have a way to slow the heart rate down. And that's going to be through the parasympathetic nervous system. Parasympathetic nervous system is going to coordinate actions, allow us to rest, relax, digest food, things like that. That activity emerges from the dorsal motor nucleus of the vagus and goes down what's known as the vagus nerve. This is a nerve that travels from our brainstem, the medulla oblongata, to many different organs within our body and allows a coordinated relaxation of many organs simultaneously. Okay? What it does is it releases a neurotransmitter called acetylcholine onto the sinoatrial and atrioventricular nodes. Okay, that inhibit them. They slow their ability for them to depolarize electrically and to trigger to slow the ability for them to trigger the electrical waves. Okay, so that will slow down the heart rate. That's called bradycardia, slowing down of the heart rate. On the other hand, we have the sympathetic nervous system, which is involved in fight or flight responses, that also emerges from the medulla oblongata. But instead of that nerve that wanders through the body, it goes straight down the spinal cord, comes out of the sympathetic chain ganglion, and then drops norepinephrine onto the heart, both at the SA node, AV node, as well as other areas of the muscle itself. That will increase the heart rate, cause tachycardia, a higher heart rate. Now, those are electrical signals that control the heart, but there are also hormonal signals that can control the heart. In particular, epinephrine, also known as adrenaline. If you get that surge, you get scared, something like car's about to hit you, your heart rate goes up, right? Suddenly, that's the actions of epinephrine that is released It's a neurohormone that's released from your brain, gets released into your body, and it's also released from your adrenal cortex on top of your kidneys, dumps it into your bloodstream. You get a sudden surge from a molecule that's circulating through the blood and hits your heart, causes the heart rate to go up. So other factors like temperature, the higher the temperature, the higher your heartbeat is going to go, higher any chemical reaction in your body is going to go. Your exercise and your body temperature goes up, so you're going to increase your heart rate. Anybody ever seen Pulp Fiction? That movie? Nobody's seen it anymore. It's classic. Tarantino, right? It's famous movie. There's a very famous scene, okay, where the girls are overdosing on heroin, right? They stab them right in the heart with some epinephrine, okay, in order to bring her back. And so that is here's the sort of physiological basis of that scene. Okay. Do one more slide. So there is a maximum heart rate. Okay. And why is there a maximum heart rate? As you increase the amount of power that you're putting out in exercise, what you'll see is that the cardiac output and your heart rate goes up. Okay. Your heart rate can only go to a certain limit though, because if you go any higher than that limit, okay, so once you start going faster and faster, your cardiac output no longer increases. And that is because, so as your heart rate's going up with more power produced, you sort of cap out your cardiac output. And that is because you don't give your heart enough time to refill. There's a peak stroke volume that you get that then starts to fall when you're beating too quickly. So that is why we have a maximum heart rate. The left ventricle can't completely fill, and beating faster does not increase the cardiac output. So that is a cap, and that adjusts with your age. Now I'll stop there. We'll continue on cardiovascular and respiratory systems on Tuesday. Thank you. Yeah.
So you put the peppermint goes into your bloodstream. Why does it take like 2-3 minutes to actually take in if your blood pumps that much blood so fast?
Oh, I think I mean, epinephrine, I think kicks in much faster, like almost instantaneously. You want this thing?
Yeah.
All right, let me get out of your way.
Tuesday, we have our midterm.
Okay.